public health officials and virologists are increasingly focused on a pathogen that remains one of the most lethal, yet frequently overlooked, threats in the natural world, the hantavirus. Often surfacing after routine domestic activities such as cleaning out a garage or organizing a shed, the virus presents a deceptive clinical picture that begins with symptoms indistinguishable from a common flu. The title of recent public health briefings, The Hantavirus Finality, underscores the gravity of the disease progression, where muscle aches following simple chores can escalate into a life-threatening respiratory crisis within hours. This report examines the current state of hantavirus surveillance and the biological mechanisms that make this family of viruses a persistent challenge for global health systems. The hantaviruses belong to the family Bunyavirales, a group of negative-strand RNA viruses that are primarily carried by rodent reservoirs. Unlike many other zoonotic diseases, hantaviruses do not rely on insect vectors like mosquitoes or ticks. Instead, they are transmitted through the inhalation of aerosolized viral particles found in the dried excreta, urine, or saliva of infected rodents. In North America, the primary culprit is the deer mouse, though other species like the white-footed mouse, the rice rat, and the cotton rat also carry distinct strains. When a person disturbs a dusty environment where these rodents have nested, they can inadvertently breathe in the virus, initiating an infection that remains asymptomatic for a period, typically ranging from 1 to 5 weeks. This long incubation period often makes it difficult for patients to link their current illness to a specific cleaning event or outdoor activity. The initial stage of Hantavirus Pulmonary Syndrome, or HPS, is characterized by the prodromal phase. This involves high fever, severe muscle aches, particularly in the large muscle groups such as the thighs, hips, and back, and significant fatigue. Because these symptoms mirror the common influenza or even physical overexertion from chores, many individuals do not seek immediate medical attention. However, clinicians emphasize that the distinguishing factor is the speed of decline, as the virus targets the endothelial cells, the cells lining the blood vessels. The body's immune response triggers a massive inflammatory event. This leads to what medical professionals describe as vascular leak syndrome, where fluid from the bloodstream begins to seep into the air sacs of the lungs. The transition from general malaise to acute respiratory distress can occur in as little as 24 hours, leaving a narrow window for intervention. Globally, the impact of hantavirus varies by geography and viral strain. While the Americas deal primarily with HPS, which carries a mortality rate of approximately 38%, Europe and Asia face hemorrhagic fever with Renal Syndrome, or HFRS. While the symptoms and target organs differ, the underlying cause remains the same: a spillover event from a rodent population to a human host. Surveillance data indicates that environmental factors, such as increased rainfall leading to higher seed production, can cause rodent populations to surge, subsequently increasing the risk of human exposure. As urban sprawl continues to push residential developments into previously wild areas, the interface between humans and these rodent reservoirs is expanding, making the simple chore of clearing brush or cleaning a rural cabin a high-stakes encounter with an ancient and unforgiving pathogen. The clinical management of hantavirus remains a significant hurdle for hospitals and diagnostic laboratories worldwide. According to official statements from health agencies, there is currently no specific cure, vaccine, or antiviral treatment approved for Hantavirus Pulmonary Syndrome. Instead, the standard of care relies heavily on early recognition and aggressive supportive therapy in an intensive care setting. Once a patient enters the respiratory phase, they often require mechanical ventilation or, in extreme cases, extracorporeal membrane oxygenation, known as ECMO. This process involves circulating the patient's blood through a machine to oxygenate it outside the body, allowing the lungs a chance to recover from the intense fluid buildup. The logistics of providing such high-level care are often complicated by the fact that many hantavirus cases occur in rural or remote areas, far from specialized medical centers equipped with ECMO technology. From a laboratory perspective, confirming a hantavirus diagnosis is a complex and time-sensitive process. Most initial screenings are performed using enzyme-linked immunosorbent assays, or ELISA, to detect specific IgM and IgG antibodies that the body produces in response to the virus. However, In the very early stages of the prodromal phase, these antibodies may not yet be present in detectable levels. Molecular testing, specifically reverse transcription polymerase chain reaction or RT-PCR, can be used to identify the viral RNA itself, but this often requires shipping samples to state or national reference laboratories. This logistical delay can be critical, as the patient's condition may deteriorate while the tests are being processed. Public health officials stress that if HPS is suspected based on the patient's history of rodent exposure and rapid onset of pulmonary edema, clinical treatment should begin immediately without waiting for laboratory confirmation. The internal mechanisms of the virus are a primary focus of ongoing research. Unlike many viruses that cause direct cell death, hantaviruses are generally non-cytopathic, meaning they do not kill the cells they infect. Instead, the severe damage seen in the lungs or kidneys is largely the result of the host's own immune system. The cytokine storm induced by the infection causes the capillaries to become highly permeable. In the lungs, this results in the sudden filling of the alveoli with plasma, essentially causing the patient to drown internally. In the Eurasian strains causing HFRS, the focus of this vascular leak is the kidneys, leading to acute renal failure and internal hemorrhaging. This distinction in pathology is a key area of study for virologists who are attempting to understand why different hantavirus strains target different organ systems with such specificity. International surveillance networks, including those coordinated by the World Health Organization, monitor these outbreaks to identify potential shifts in viral behavior. While person-to-person transmission is extremely rare for most hantaviruses, an exception has been documented with the Andes virus in South America. There, small clusters of cases have suggested that the virus can spread between close contacts, a development that has significantly altered the public health response in the region. For health agencies, the priority remains the verification of data from clinics and the maintenance of rigorous testing standards. The disparity between claims of new cures and the verified reality of supportive care is a constant point of friction, with officials urging the public to rely only on evidence-based medical guidance from established institutions. Prevention and public guidance remain the most effective tools in reducing the incidence of hantavirus infection. Because the virus is so closely linked to the presence of rodents, health departments emphasize a no-contact approach to rodent management. The primary recommendation for anyone tasked with cleaning areas where rodents may have nested is to avoid any activity that might kick up dust. This means strictly avoiding the use of brooms or vacuum cleaners in garages, sheds, or cabins that have been closed for a season. Instead, the protocol involves thoroughly wetting the area with a disinfectant or a mixture of bleach and water. This wet cleaning method prevents viral particles from becoming airborne, which is the primary route of infection. Wearing gloves and, in some cases, high-efficiency particulate air masks, commonly known as N95 respirators, is also advised for those working in high-risk environments. Surveillance programs are currently attempting to predict hot years for hantavirus by monitoring environmental indicators. Scientists have noted a correlation between the trophic cascade, where high rainfall leads to a surplus of food for rodents, and subsequent spikes in human cases. By tracking these ecological shifts, public health agencies can issue early warnings to specific regions, advising residents to be extra vigilant about rodent-proofing their homes. This involves sealing even the smallest cracks or holes in foundations, keeping food in rodent-proof containers, and removing debris piles from near the home. Despite these efforts, the limits of current knowledge are clear. It is still not fully understood why some individuals exposed to the virus develop severe disease, while others might have a more muted response. Though genetic factors and the initial viral load are suspected to play significant roles, the reality of hantavirus reporting is one of caution and ongoing vigilance. While the numbers of cases globally are relatively low compared to more common respiratory illnesses, the high mortality rate and the sudden nature of the illness make it a significant public health priority. There is an honest limit to what can be done once the late-stage respiratory failure sets in, which is why the focus remains so heavily on the early warning signs. That specific combination of high fever and muscle pain following domestic chores. there is no room for complacency in regions where the rodent reservoirs are known to exist, and health officials continue to refine their outreach to ensure that both the public and local physicians recognize the symptoms before the disease reaches its final, critical stage. In conclusion, the hantavirus serves as a stark reminder of the complex relationship between human activity and the natural environment. As we continue to share spaces with wildlife, the importance of basic hygiene, informed prevention, and rapid medical response cannot be overstated. Health agencies will continue to monitor the situation, from the molecular level in high-containment labs to the ecological level in the field, seeking to mitigate a threat that remains as silent as it is deadly. For more updates on global health surveillance and emerging viral threats, consider following our ongoing reporting on these critical issues. The clinical progression of hantavirus within the hospital setting is characterized by a deceptive calm followed by a rapid, often catastrophic physiological decline. Medical facilities reporting on these cases highlight a specific diagnostic challenge: the early symptoms, high fever, severe myalgia, and gastrointestinal distress, provide no immediate signature of the virus. However, as the patient moves toward the critical phase, hematological markers begin to shift. Laboratory clinicians look for a specific triad of findings: a significant drop in platelet counts, known as thrombocytopenia, an increase in white blood cell counts with a high percentage of immature or atypical lymphocytes, and an elevated hematocrit level, which indicates that the liquid portion of the blood is leaking out of the vessels and into the surrounding tissue. This hemoconcentration is a primary indicator that the patient is entering Hantavirus Pulmonary Syndrome, or HPS. Unlike many viral infections that cause localized tissue death, the hantavirus targets the endothelial cells that line the entire circulatory system. This interaction does not kill the cells directly, but instead triggers an overwhelming inflammatory response. This inflammatory event, often referred to in medical literature as a cytokine storm, results in the sudden permeability of the capillaries. In the lungs, this manifests as a massive influx of plasma into the alveolar spaces, essentially obstructing the exchange of oxygen. According to data from the Centers for Disease Control and Prevention and various international health ministries, once this respiratory phase begins, the survival window narrows significantly. In regions across the Americas, where the Sin Nombre and Andes virus strains are prevalent, the mortality rate consistently hovers near 38 to 40%. The intensity of this transition is why public health officials emphasize that any history of rodent exposure followed by flu-like symptoms must be treated as a medical emergency. Contrast this with hemorrhagic fever with Renal Syndrome, or HFRS, seen more frequently in Europe and Asia. In those cases, the vascular leak primarily affects the kidneys, leading to acute renal failure and internal bleeding. While the target organ differs, the underlying viral mechanism of vascular dysfunction remains the core development that medical teams must combat. The diagnostic timeline remains a point of logistical friction between local clinics and specialized laboratories. Because hantavirus is a biosafety level 4 pathogen in its concentrated form, many standard hospital labs are not equipped to handle the virus itself. Instead, they rely on serological testing to detect the presence of antibodies. The enzyme-linked immunosorbent assay, or ELISA, is the standard for identifying IgM antibodies, which appear early in the infection. However, because the viral load can be low during the initial prodromal phase, a negative test result in the first few days of illness does not definitively rule out the virus. For a more definitive confirmation, samples are often dispatched to state or national reference laboratories for reverse transcription polymerase chain reaction, or RT-PCR testing. These labs look for the specific genetic sequences of the hantavirus RNA. The delay inherent in shipping these samples from rural locations, where the majority of cases occur, to centralized urban laboratories creates a gap where clinicians must make high-stakes decisions based solely on the patient's rapidly deteriorating physical state. Regarding treatment, there remains a stark absence of an off-the-shelf antiviral cure. While ribavirin has been used in clinical trials for HFRS in Eurasia with some success, it has not shown a clear benefit for the pulmonary syndrome found in the Western Hemisphere. Consequently, the medical response is entirely supportive. For the most severe cases, hospitals utilize extracorporeal membrane oxygenation, or ECMO. This system acts as an external lung, pulling blood from the body, removing carbon dioxide, and pumping it back in with fresh oxygen. This allows the patient's own lungs to rest and potentially recover from the inflammatory fluid surge. However, the availability of ECMO is extremely limited, often restricted to major university hospitals or specialized cardiac centers. The logistics of transporting a patient in acute respiratory distress via air ambulance to an ECMO-capable facility adds another layer of complexity to the public health response. International agencies continue to verify reports of new experimental treatments, but for now, the consensus remains that early detection and aggressive fluid management in the ICU are the only verified methods to improve a patient's chance of survival. Public health guidance for hantavirus prevention is centered on a single critical objective: preventing the aerosolization of viral particles. Because the virus is excreted in the urine, saliva, and feces of infected rodents, any activity that creates dust in an infested area can launch these pathogens into the air. Health agencies across the globe have issued standardized protocols for what is known as the wet cleaning method. This technique explicitly forbids the use of brooms or vacuum cleaners in areas like sheds, cabins, or crawl spaces that show signs of rodent activity. Instead, the standard procedure requires individuals to thoroughly saturate any nests, droppings, or contaminated surfaces with a disinfectant solution, typically a mixture of 1 part bleach to 9 parts water. By allowing this solution to sit for at least 5 minutes, the disinfectant can penetrate the organic material and neutralize the virus's lipid envelope, rendering it inactive. Only after this thorough soaking should the material be wiped up with paper towels or rags, which must then be sealed in plastic bags and disposed of immediately. Practical takeaways for homeowners and outdoor workers extend beyond cleaning to the structural fortification of dwellings. Known as rodent-proofing, this process requires a detailed inspection of the building's exterior. Surveillance teams note that mice can enter through openings as small as 1/4 of an inch, roughly the diameter of a pencil. Effective prevention involves sealing these gaps with materials that rodents cannot easily chew through, such as heavy-duty steel wool, held in place with caulking or expanding foam. Furthermore, environmental management near the home is vital. This includes moving wood piles away from the foundation, keeping grass mowed short to remove cover, and ensuring that all pet food and trash are stored in airtight, heavy-duty plastic or metal containers. For those working in high-risk environments where ventilation is poor, the use of a NIOSH-approved N95 or P100 respirator is not merely a suggestion, but a critical safety requirement to filter out microscopic viral particles. The limits of our current knowledge regarding hantavirus remain a source of concern for the global scientific community. While we understand the transmission route and the primary rodent reservoirs, we do not yet fully comprehend the variability in individual immune responses. It remains unknown why 2 people exposed to the same environment can have vastly different outcomes, with one experiencing mild flu-like symptoms and the other progressing to fatal pulmonary failure. Furthermore, while the majority of hantaviruses are not known to spread from person to person, the Andes virus in South America has demonstrated this capability in specific clusters. This potential for mutation or behavioral shifts in other strains keeps international surveillance teams on high alert. Researchers are also still investigating the long-term health impacts for survivors of Hantavirus Pulmonary Syndrome, as the intense scarring and physiological stress of the respiratory phase may leave lasting effects on lung capacity and cardiovascular health. Ethically, the reporting on hantavirus requires a careful balance between public urgency and the avoidance of undue panic. Public health officials emphasize that while the virus is exceptionally lethal, it is also relatively rare and entirely preventable through informed action. The ethical responsibility of the individual lies in recognizing the risk of simple chores and taking the extra 10 minutes to follow proper safety protocols. For the medical community, the duty involves maintaining a high index of suspicion during the prodromal phase,